Good morning, students. Myself, Major Komal Katyal, Department of Zoology, G.K. Govani Government College, Bhinmal. In continuation with my previous lecture on cells of the immune system, we will begin today's lecture. Now here in this slide, various types of WBC, which are popularly known as white blood cells or white blood corpuscles, are shown. Out of these white blood corpuscles, we have already discussed about B lymphocytes, T lymphocytes, and natural killer cells in my previous lecture. Today we will discuss about the monocytes, which are short-living and they migrate to the tissues and become metamorphosed into macrophages. Then the three different types of granulocytes: neutrophils, eosinophils, and basophils. And these basophils later on becomes the mast cell responsible for causing inflammation. Now first we will discuss about the monocytes. The monocytes are circulating immature cells of mononuclear phagocytic systems. There are two type of system, one the mononuclear phagocytic system and other the polymorphonuclear phagocytic system, which includes the neutrophils. Monocytes are basically 10 to 20 micrometer in diameter and they consist of approximately 4 to 5% of the circulating WBC. Their azurophilic lysosomal granules contain lysozymes, myeloperoxidases, and acid hydrolases. As I have told you earlier on, they have a short half-life which is of about 24 hours only, and these monocytes later on migrate to the tissue and then they metamorphose or become the macrophages, as shown in the slide also, that they migrate to lungs, bones, liver, brain, and so on. Now how this phagocytosis takes place is depicted here in this slide. First of all, the pseudopodia surround the microbes. Then the microbes are engulfed into the cells and a vacuole is formed which contains the microbes. After the vacuole formation, the vacuole plus primary lysosome fuse and thereby there is formation of secondary lysosome. The toxic compounds, acid hydrolases, and other lysosomal enzymes destroy the microbe, and the dead remains of the microbes are removed from the cells through exocytosis. The macrophages. As I told you earlier on, the macrophages are metamorphosed monocytes which occur in the tissues. So they are the differentiated monocytes which migrate from blood to the tissues. Unlike monocytes, they have a long lifespan from many months to even years. They are the big eaters of the tissues since neutrophils are present only in the blood. They are responsible for carrying out the phagocytosis of microbes in the tissues. More densely present in the spleen, but their number is more in the liver because liver has bigger size than the spleen. The Kupffer cells and the histiocytes of the liver are common example of macrophages. Alveolar macrophages and peritoneal macrophages are common example of tissue macrophages present in the lungs. The microglia in the brain are also example of the macrophages. Giant cells of epithelium are also macrophages. So the function of macrophages include phagocytosis, processing of the antigen, presentation of antigen, and in the last they are responsible for secretion of certain hydrolytic enzymes. Now about the granulocyte, first of all about the neutrophils. They are the most abundant WBC, constitute approximately 40 to 70% of circulating white blood cells. They are efficient phagocytes and they are the polymorphonuclear leukocytes. Most important cells of the immune system, these neutrophils find, eat and digest the microbes. They are motile, found circulating in the blood, and they are the short-lived cells. Their lifespan is only 2 to 3 days, although it takes about 6 to 9 days to develop them. Their size is approximately 10 to 20 micrometer in diameter. Now, like monocytes, the neutrophils are also responsible for carrying out the phagocytosis, and like monocytes, they are also responsible for first forming the pseudopodia, engulfing the microbes, thereby forming a phagosome which combines with the lysosome, and thereby formation of secondary lysosome, and so on. Ultimately, after the lysis of microbes, the debris or the remains of the microbes are discharged from the cells. As far as the additional role of neutrophils are concerned, they are responsible for carrying out certain additional function, one of which they are responsible for triggering the inflammatory response. As we have seen commonly, that whenever a body part is injured but not wounded, it becomes swollen, and sometimes when an insect bites, the inflammation occurs. So this inflammation is actually due to inflammatory response which is triggered by the neutrophils. The second one, the eosinophils. The eosinophils are basically the acidophilic polymorphonuclear leukocytes, which are also known as acidophils because they absorb the acid dyes. Their nucleus is usually bilobed or ellipsoid. Their size varies from 2 to 5 micrometer in diameter. Their lifespan varies from 8 to 12 days, and they constitute about 2 to 5% of circulating WBCs. They release histaminase and aryl sulfatase, which are the enzymes which inactivate the mast cells products histamines and leukotrienes. They slow down the process of inflammation, means antagonistic to the mast cells. They play an important role in the immunity to the helminth parasites or the worms. They also destroy the antigen antibody complexes. Now, the basophils and the mast cells. The basophils are the basophilic polymorphonuclear leukocytes. Their nucleus is usually bilobed. The size varies from 2 to 5 micrometer in diameter. They constitute about 0.5% of the circulating WBCs. Their lifespan from few hours to few days, and the main function is release of histamine and other mediators of inflammation. They also contain heparin, an anticoagulant which is generally present in our blood and prevents the coagulation or thrombosis of the circulating blood. The basophils modifies in the form of mast cells, and these mast cells are large tissue cells with basophilic granules. The following are the examples of mast cell: the mucosal mast cells, which is present in the mucosa; connective tissue mast cells, the mast cells which are present in the connective tissue, and these mast cells, like basophils, mediate the inflammatory or allergic reactions. Now, then the APC or the antigen presenting cells. The antigen presenting cells are basically non-lymphocytic cells whose main function is to carry the antigen and present them to the immunocomplement lymphocytes, which are located in the secondary lymphoid organs. There are different types of antigen presenting cells which are present in our body. First of all, we will discuss about the follicular dendritic cells. The follicular dendritic cells are found in the germinal centers of the lymph nodes and the spleen, and they possess the Fc and C3 receptors, but they lack the class second major histocompatibility complex or class second MHC. The second kind of antigen presenting cell or APC are interdigitating dendritic cells. They are more or less like the follicular dendritic cell, but the basic difference is they are found in the paracortex of the lymph nodes, while the follicular dendritic cells were present in the centermost region or the medulla region. They possess class II glycoproteins but lack Fc and C3 receptors. Their structure similar to the follicular dendritic cells. If we look at the dendritic cells, they are more or less, they have a structure more or less similar to the dendron or dendrite of a neuron or a nerve cells. And then the Langerhans cells. The Langerhans cells were the first antigen presenting cell discovered by the immunologist, and they are named so because they are found in the islet of Langerhans in the pancreas. They possess class 1, class 2 MHC or the major histocompatibility complex. They also possess Fc and F3 receptor. It means they possess all type of receptors and carry out the presentation of antigen more efficiently to the antibody secreting plasma cells. This finishes our lecture on cells of the immune system. Thank you.